Okay, so I'm going live here. I don't expect too many people to be on right now. Have to do this when I can. It turns out my wife had to go into work today, so I've got the kid. However, it's week 8, and now it's time for Bio 1140 Test 2. Now, this is by far one of the hardest tests you'll ever take. And a lot of students are like, why is a test in an 1100 class so hard, or a freshman level class so hard? Well, it's all in context, right? It's if you were in 201 or 202 or 304, it wouldn't be that hard for you. Or if you were in a nursing program, or even if you were in med school, you'd be like, I remember those good old days of 1140 when they were easy. What makes it hard is for many of you, you've never really seen this material before, so it's really new. And because you're learning a lot of new material all at once, that makes it hard. And also trying to understand the material rather than just memorize a bunch of definitions that that you can easily Google and look up. And that's what makes this really hard, because like I said, it's a more of an intellectually challenging test than what you're used to, especially coming out of high school. Now for college level, it's not so bad. Hold on one second here. Yeah, you caught me drinking a Rockstar. What can I say? Okay, so I did get a few questions for this test. So I'm going to go over these really quick. And the first one is, let's see, about phospholipids and cellular membranes and how it's amphipathic, both hydrophobic and hydrophilic. But how does this pertain to cellular membranes? That's actually a really great question. And here's why. The fact that a cellular membrane has a hydrophobic interior. Now, hydro means water, phobic means fear of. So what that means is the interior of a membrane is hydrophobic. It doesn't like water, and it doesn't like anything that has partial charges to it that's soluble in water. It doesn't like electrolytes like sodium, potassium, chloride. These things don't pass those membranes very well. Now, the outside of a membrane is hydrophilic because there's a phosphate head attached to that. So what that means is there's a hydrophilic part of the membrane, which is outside, of the interior. And basically that's attracted to water, but the interior part is hydrophobic. That is very important because what it does is it sets up something called selective permeability. And I'm going to show you really quick what a membrane looks like. If we can draw one really quick here. I never went into art school for a reason, but this is a cellular membrane. These are the hydrophilic heads right here, like this. And this are the hydrophobic fatty acid tails. And of course, my fancy whiteboard is, of course, not with me right now, so I'm having to use some paper here. But these two tails represent fatty acids. And if you remember what a fatty acid looks like, they have a carboxyl group at one end, followed by a long chain of carbon. So if you're looking at this and you're like, I don't understand what that line is. You don't understand that line is a covalent bond, and this node right here is a carbon, right? So this right here would look like this. There's a carbon, that's that carbon. There's some hydrogens coming off of here, some carbons here. So this is a fatty acid tail. There's my carboxyl group right there. And this is hydrophobic because all this carbon and hydrogen, these are nonpolar covalent bonds, something you learned in chapter 3. These nonpolar covalent bonds, well, they don't interact with water in any way, right? They can't form any hydrogen bonds. So as a result, these hydrophobic tails, they just don't let water or anything like an ethanol or an amino acid or electrolytes pass through them. So what that means is membranes have a property called selective permeability. And I'll write that down here, selective permeability. What selective permeability means is that only certain things can cross the membrane. So when you're breathing in oxygen, oxygen is O2. It can go right through the membrane, no problem. When your body creates carbon dioxide through cellular respiration, CO2 can leave right through the membrane, no problem. Now, water, being small, can actually enter and exit a cell, although more slowly than carbon dioxide or oxygen can. But if you get something like an electrolyte, sodium, potassium, chlorine, those are ions, they have charges, and they will not cross the cellular membrane because the cellular membrane has selective permeability due to that hydrophobic interior. One more thing about the molecule being amphipathic, because the hydrophobic fatty acid tails don't like water, they turn in toward each other like this to get away from the water that's out here, whereas these hydrophilic heads, if you remember, the hydrophilic heads are made up of phosphate groups. So a fatty acid tail looks something like this, which I'll draw for you really quick. And I ran out of space. I should have gotten a bigger piece of paper here. This is a phosphate, this is horrible drawing, but this is basically my phosphate head, and then this is my glyceride, and these are my fatty acid tails right here. There's my carboxyl groups that you see right there. So this is, of course, totally hydrophobic. We can take a phosphate group, and we can see that these oxygens are really electronegative compared to the phosphate, and as a result of them being very electronegative, they can form hydrogen bonds with water or other forms of electrostatic attraction. You know, electrostatic sounds like a big word, but if you've ever played with a magnet, you know that plus and minuses attract. So the hydrophilic heads are phosphate heads, and they of course are interact with water, and that's why they turn out to the outside, and the hydrophobic heads turn out to the inside. That means that membranes can form easily on their own. So cells really don't have to do a lot of work assembling a membrane in terms of getting those hydrophobic tails to interact with each other. Okay. I hope that explains why that's important. And like I said, every cell on this planet is surrounded by a membrane that has that selective permeability. And that's really important because, as I told you, we can't really control pump water in or out of a cell. Water just diffuses across the membranes. Now, we do have proteins called aquaporins that will allow water to leave a cell rapidly. You find those in your kidneys where you have to process lots of water. So if you're like me drinking a rock star here, you're processing lots of water. But if you want to bring water in a cell, your cells then have to do what is called active or passive active or facilitated diffusion, active transport or facilitated diffusion to move ions into the cell that changes the water potential, bringing water in the cell. If you have too much water inside the cell, then you're going to remove electrolytes out of the cell, and then water will move out as well. So the way we actually control water balance inside of our cells is by actively transporting electrolytes either into or out of the cell. And kind of going off on a tangent here, but that's really important for things like understanding diarrhea. We've all traveled, we've all gotten sick. You get some diarrhea. Yeah. What's happening there is your cells aligning your intestines while they regulate ions, which regulates water uptake in your body. If you get diarrhea, you've got some pathogen inside your intestines that have affected its ability to regulate ions. Basically, you've opened up all the ion channels, and all your electrolytes, which are ions, dump out of your body into your intestine. You lower the water potential because all your calcium and sodium and potassium and chlorine are going into your intestines. Water flows out of your body into your intestines. It fills up, and you're into the bathroom. So that's why these things are important. Okay. Another question I got, and this also exemplifies, some of the difficulty with this class as well. The question was, how does a swimming pool, an Olympic swimming pool, have more kinetic energy than a hot cup of coffee? Here it is. Coffee, a hot cup of coffee got, not very much. It's a few ounces of water in there. It's got a higher average kinetic energy because it might be, 130, 140 degrees Fahrenheit. So it's got a higher average kinetic energy than, let's say, an Olympic swimming pool, which is around 80 degrees. I know that sounds warm, but 80 degrees is kind of cold if you're in the water. But that being said, the average is higher in a cup of coffee. Now, here's the difference. The swimming pool has way more total energy. Here's how you know. Let's say I took a hot cup of coffee at 140 degrees and an Olympic swimming pool 80 degrees. I put them outside where it's 20 degrees. That coffee will freeze within hours. Within an hour, actually, it'll freeze. The Olympic swimming pool might take several days to lose all that energy stored inside of it to start to cool off. In fact, it might take weeks for an Olympic swimming pool to freeze, going from 80 degrees to 20 degrees, because there's so much more kinetic energy in the pool. So the total amount of energy is much higher, just that the averages are different. So those are two of the questions that I've gotten for this test. Now, here are my study tips for this. As you realized on the first test, you are not going to be able to simply Google those questions and get simple definitions and get answers out of it. You have to understand what the material is. So for instance, a good way to understand material, memorize things like this. This is our basic chemistry. This is some basic organic chemistry that are needed for this class. But I've got lots of videos on this. You need to know things like, okay, if I have carbon, hydrogen, oxygen, and hydrogen, you should be able to look at this molecule right here and say, okay, carbon and carbon have the same electronegativity. They're going to attract electrons about the same. Same with hydrogen here. These are nonpolar covalent bonds. They're not going to form hydrogen bonds. If we look at something like carbon and hydrogen and oxygen, this of course is a carboxyl group. Oxygen is more electronegative than carbon. That means the electrons are over here. That means that the electrons spend more time near the oxygen, so it's partially charged negative, but it's pulling the electrons away from the hydrogen and is partially positively charged. And as a result, this molecule will form a hydrogen bond with water. Now, here's the thing. If you don't know what electronegativity is or why oxygen is more electronegative, then you need more understanding. You need more knowledge about, like, oh, oxygen has 8 protons. Carbon has 6 protons. Protons are positively charged. If I've got 8 protons, I've got more positive charges than I do in the carbon. So I should be able to draw any random molecule and say, hey, is this going to be hydrophobic or hydrophilic? Which parts of it will be hydrophobic and hydrophilic? You can do that by understanding basic atomic structure. Also, the macromolecules. Okay, this is what gets people every single semester. I mean, it gets them. I mean, macromolecules. And what I discovered is that people look at them, and they look at a glucose molecule, and they look at an amino acid, and they look at a nucleotide, and they're like, oh yeah, I got this. I got this memorized. You see it on a test, it's a slight variation. You become familiar with your notes. You haven't really understood what it means to be a carbohydrate or an amino acid. And of course, if you're wondering what an amino acid is, I can draw one right here. For all you Star Trek fans, you start with NCC, nitrogen, carbon, carbon, Naval Construction Contract 1701. That's a USS Enterprise. Out here, we have a hydroxyl group, central carbon, and amine group on the end. So there's our amine group. There's a carboxyl group. And anytime you see this, this is an amino acid. The best way to figure out the amino acids, which are building blocks of proteins, is to draw it. Draw it over and over and over. You want to draw an amino acid? Now you know what it looks like. You want to draw a carbohydrate? A carb, sorry. You know, I'm just drawing this off the top of my head here. Draw a glucose molecule. So when you have to see this on a test, oh, H, gosh, I can't draw here. H, O, H, O, H. I'm adding in hydroxyl groups. There's a carbohydrate. This is glucose right here. And the thing about it is, why does sugar dissolve in water? Here's understanding once again. Glucose has lots of hydroxyl groups. There are 5 hydroxyl groups. Each one of these hydroxyl groups can form a hydrogen bond with water, and therefore you can get 140 grams of sugar inside a single soda can, because this is highly hydrophilic. Hydro meaning water, phile, philic meaning loving, because it can form hydrogen bonds. And of course, this is a simple carbohydrate. This is an amino acid. Learn to draw these things. That's how you get, that's the basics of getting past those macromolecules. You have to just learn how to draw them. Also, you know, we always remember the simple things. We come into the class knowing carbs are used for energy. Why are they used for energy, right? And what else are they used for? You know, carbs are way more important than just being burned off for energy. We use them as we break them down and use them for building blocks for other things. And plants, cellulose is the most common molecule, organic molecule on the planet, probably. Cellulose is, of course, what makes wood. It's used for structural support. And if you've ever heard of like A, B, and O blood systems, those are glycoproteins. Glyco meaning sugar, protein. Those are glycoproteins on the surface of our cells. So carbs are also used for cell recognition. That's how your body recognizes itself from other entities or even certain tissue types. So carbs are very, very important. And of course, you need to memorize that. That way, if you see something about what they're doing, you'll get it. The next chapter is on cells. That chapter is, you just got to memorize the parts of a cell. I mean, that's just basic biology. A lot of you go, man, I'll never see this again in my life. And you're right. You might not. But it's also, never forget that we're teaching you a little bit about this world, about our basic bodies, about how, you know, cells are the basic unit of life, and understanding a eukaryotic cell, which makes up the 17 trillion cells in your body, is kind of interesting, right? And it has direct impact into your lives in terms of like fertility. Amazingly, if your mitochondria aren't working right, you have low energy, you might be infertile. That's why we have some kids out there now that they're doing these experiments with 3 parents. You got a sperm donor, an egg donor, and a mitochondria donor, right? So understanding mitochondria is incredibly important for understanding things like fertility, metabolic issues, how your cells commit suicide. Yeah, your cells actually commit suicide, and that's involving with the mitochondria. Aging has to do with mitochondria. We learned about things like lysosomes. Oh yeah, you know, lysosomes, they go around, they digest other organic molecules, but they also digest your organs. And part of what happens as we age, the lysosomes no longer eat your organelles, eat other organelles, turn them over like mitochondria. And the slower we turn over those organelles, our cells accumulate damage, and that's part of our aging process. And learning how we could reset the clock on like something as simple as autophagy, self-feeding, getting our lysosomes to turn over our organelles faster like they do in a young person, can reset the clock on aging. So yeah, these things are incredibly important. Okay, I've got another question. Ashley Martinez, what is the importance of functional groups? How do we differentiate between structures of functional groups and complex structures such as glucose molecule? Fantastic question. So a functional group are things like a hydroxyl group, which is an OH, or a carboxyl group, which is something that looks like this right here. That's, of course, a C attached to carbon and oxygen and hydrogen. A carbonyl group. Of course, this is a carbon double bonded to an oxygen right there. Carbon double bonded to an oxygen and hydrogen for hydroxyl. This is just a hydroxyl right there. And of course, something like an amine group. So functional groups are small groups of molecules that we attach to other molecules that change their properties. So here's a great example of how a functional group works. We can take a natural gas, what we burn, not one that you might produce in your gut, but one you might get out of the ground to burn. This is ethanol. This is a gas. It is highly flammable. You have to put it under pressure, cool it down to make it a liquid. But this is a gas. This is what we burn as natural gas. This is one of the components of it. Let's say I add on a functional group to ethane, and I add on the functional group hydroxyl. There's a hydroxyl group right there added to my ethanol. So I went from ethane to ethanol. Now, many of you know about ethanol. Maybe you might try some ethanol on the weekends, only if you're 21, or if you're in every other country that's civilized, you can drink earlier. But basically, this is a gas at room temperature. This is a natural gas. We burn it in our stoves. Then we put this in our drinks. This is a liquid at room temperature and easily dissolves in water. And the reason why is of this hydroxyl group. This is a hydrophobic molecule. It is full of carbon and hydrogen. The electronegativities are roughly the same. This cannot form a hydrogen bond with water. It does not really form hydrogen bonds, so it is hydrophobic. It is water-fearing. This molecule is hydrophilic. The reason why is because of this hydroxyl group. Oxygen is way more electronegative than hydrogen or carbon. So as a result, the oxygen attracts all the electrons near it, and it becomes partially negatively charged. This hydrogen becomes partially positively charged. So you stick this in water, and it forms a hydrogen bond with water. So that's one way that a functional group changes the properties of a molecule. We can also look at our amino acids. Amino acids are considered to be hydrophobic or hydrophilic, depending on their side chain. Now, of course, this right here, this NCC, R is just a side group. This is the backbone of the amino acid that links together to form a protein, but this represents a side group. If I put a side group on here that's just full of carbon and hydrogen, like this one long branch right here, this would be a hydrophobic amino acid because this is incapable of forming a hydrogen bond with anything. However, if I add on a hydroxyl group or let's say a carboxyl group down here, that would only, of course, have no hydrogens at this point. This carbon can only form 4 covalent bonds. Now all of a sudden, now my amino acid is now hydrophilic because this carboxyl group could form hydrogen bonds with water. So that's how functional groups work. So if we were to look at a glucose molecule, I think you came in after I drew this. I took the backbone of a glucose and water. If I drew something that's just carbon, 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 carbon like that, this would be mostly hydrophobic, but I add on these functional groups, these hydroxyl functional groups. Now I have a carbon, hydrate, having to do with hydrogens in there. Oh, we need one more hydroxyl group right there. Now, this molecule went from being hydrophobic to completely hydrophilic, and as a result, these functional groups really do matter. Now, also in sugars, we can draw a sugar out of, well, we can draw glucose there. Let's see here. Let's draw fructose. Fructose looks a little bit different than glucose. Right? And if we draw fructose here, fructose is what we call an isomer of glucose. So they both have the molecular formula C6H12O6, like this. This is the chemical formula. It's got 6 carbons, 12 hydrogens, and 6 oxygens. Both of these molecules have that, but these are isomers. This forms a 6-membered ring structure. This forms a 5-membered ring structure, but there's still 6 carbons here. And the reason why this is different is because of the functional groups, the position of the carbonyl group, which it could either be terminal, 1, 2, 3, 4, 5, 6. So I'll do both of them here in linear form. 1, 2, 3, 4, 5, 6. And the difference between glucose and fructose is the position of this carbonyl group on this carbon chain, which is when it forms here, it does something a little bit different than here. So the position of that functional group makes a difference between glucose and fructose. And both sugar, don't want to eat a lot of sugar in your diet. That's why this is diet. Got like 10 calories or something. But they are treated a little bit different in your body. For one, glucose causes insulin to be released, which causes it to get brought into your cells. Fructose is hauled off to your liver where it's broken down from there. Okay, actually, you got another one. What is an organic compound? You know, that's a fantastic question. An organic compound is anything that has carbon and hydrogen into it. So think about it. If I've got, so all these molecules I've been drawing here, carbon and hydrogen, that makes an organic compound. Ethane, ethanol, glucose, all your 4 macromolecules, proteins, carbs, lipids, nucleic acids, all organic compounds. All of your vitamins too, like vitamin B, vitamin C, vitamin D, vitamin K, all of these are organic compounds. What's not organic is carbon dioxide, CO2. Even though it's got carbon in here, this is not considered an organic molecule because there's no hydrogen attached to the carbon. So anytime you've got carbon attached to at least one hydrogen, you've got an organic compound. Okay, another one from Ashley. Okay, great. The 4 levels of protein structure. Hang on. You get a little bit of a head cold over the weekend. Your daughter gets sick, your wife gets sick, then you get sick. So throat's a little scratchy here. Okay, there are 4 levels of protein structure. And you can just do primary, secondary, tertiary, and quaternary. So we got, think of it this way, primary, secondary, tertiary, and quaternary. That's a 4. I don't draw very well, sorry about that. That's a 4. Primary structure. Now we can talk about primary structure of proteins, primary structure of a complex carbohydrate, and primary structure of DNA and RNA, the nucleic acids. Here's how it works. The primary structure of a protein is the sequence of amino acids linked together. So there's like these 20 amino acids, 20 amino acids, not adenine, sorry, like glutamine, glutamine, aspartic acid, glutamic acid, these things. There's 20 of them. You string them together. That is a primary structure of a protein. Primary structure of a carbohydrate would be just linking a bunch of glucose molecules together. Primary structure of DNA is something you're very familiar with. There are 4 bases of DNA: guanine, adenine, thymine, cytosine. And so the primary structure of a nucleic acid are the sequences of nucleotides. So GATTACA, guanine, adenine, thymine, adenine, cytosine, adenine. That would be the primary structure of DNA. So anytime you see primary structure of these macromolecules, it has to do with just linking the building blocks together. Oh, and by the way, Gattaca, if you haven't seen Gattaca, go see it. Fantastic, fantastic movie from the 90s. And we're basically at the point in our technology where, yeah, we can start engineering people. That's scary. That's a whole nother topic to talk about, though. But we're basically getting there. Okay, so that's primary structure. Now, secondary structure has to do with the folding of the backbone of these structures. So if you notice something like amino acids here, you notice there's some nitrogen attached to hydrogen. That's a that's a polar covalent bond. So secondary structure, basically what's happening is this backbone is going to interact with other members of the backbone and cause it to fold. So you might take it and it might start to fold like this. And this would be the backbone. We're not worried about the side chains, but there's hydrogen bonds going to form that together. That would be secondary structure of a protein. And in secondary structure of DNA is, if you remember your base pairing, guanine always pairs with cytosine and thymine always pairs with adenine. So what holds like DNA together, guanine pairing with cytosine and thymine pairing with adenine, these are held together by hydrogen bonds. That would be secondary structure holding these two things together. Now, every molecule has tertiary structure. Tertiary structure is your 3-dimensional structure of your molecule. That is, what is its length, its width, and its height? What is its overall shape? In a protein, the tertiary structure of a protein is determined by interactions of these side chains right here. Now, R is just represent there's something down here. And there's 20 different side chains used by life. They can be hydrophobic, they're hydrophilic, but the tertiary structure of a molecule is how it actually folds together to form a functional molecule based on interactions of these side chains here. That's tertiary structure. In DNA, the tertiary structure of DNA is a double helix. It's that twisted ladder. And then quaternary structure is, think of things like hemoglobin, which carries oxygen in our blood. Hemoglobin is actually made up of what we call 4 polypeptide subunits. Now, polypeptide, poly means many. Peptide has to do with these amino acids. So you can take 4 different strings of amino acids and put them together, and you get multiple polypeptides, and you put them together into one unit, like a hemoglobin molecule. And that would be the quaternary structure of a protein. Not all proteins have quaternary structure, but all of them do have primary, secondary, and tertiary structure. Just some of them, like I said, have this additional layer of complexity, but that's due to having multiple of these polypeptide strands hung together. Okay. Anybody else have any other questions while I'm still here? I think we're almost there. Lastly, the last chapter is on membranes and energy. And you need to know those structures of the membrane. I just talked about it earlier. You know, why does selective permeability matter? What's the relationship of membranes and transport? Why is it so important that membranes have selective permeability? And I remember it has to do with water, right? We control our water in our cells by controlling electrolytes. Muscle contractions controlled by electrolytes. Nerve conductance, that's electrolytes crossing membranes. So a lot is important about membranes. I mean, they are the gatekeeper. So incredibly important. Also, energy is very important as well because all life needs energy to exist. And the vast majority of energy on this life, the vast majority of energy used by life is transformed to ATP across membranes. So membranes are also important in making energy available to cells. And that's why I always say the secret to life is chemiosmosis, which is something we're going to talk about in chapter 7. It's one of my favorite things to talk about, and I'll have some more videos for that. Okay, if we don't have any other questions, it's been about 30 minutes right now. Good luck on you guys taking your test. And, I'm going to do you guys a favor. I know it's fall break. I just realized it on online class. I don't know how much that matters to people, but I'm going to go ahead and open up the test in the next hour or so. I'm going to just double check it, and you guys will actually have a few extra days to take the test. Still, you'll have one chance, 60 minutes, but I'm going to give you can take it so you don't have to take it the weekend. All right, study hard, and I'll see you next week.